and different stages of the analysis are performed in different sections on this multi-omics platform. So first of all, let's begin by signing up on the portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, OrcID, or even Twitter. A brief bio about yourself that will consist of your education background and research interest. The mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. And finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the Omics Logic Learn portal. I'm sharing the link in the chat box. Please click on the link and I'll walk you through the process behind signing up on the portal. So here is the link. All right, so for users who do not have an existing account on the portal, please click on the Create an Account option that you see here. Here you will be asked to enter your name, email ID, and provide a new password. And you also have the option to sign up using any of your social media accounts. That could be your Google account, Facebook account, Apple account, GitHub account, or even your Twitter. So once you have signed up, please put a yes in the chat box. I'll wait for a couple of minutes so that participants can sign up on the portal. Since today's session is going to be hands-on, it would be great if you could create an account now itself so that you could get access to the resources on the portal as well. So once you're done signing up on the portal, please put a yes. I'm monitoring the chat as well. Or if you're facing any technical issues while signing up on the portal, please put them in the chat as well. And our team will be happy to help you. All right, sure. I'll pause for a couple of moments so that you can sign up. So for those of you who already have an account on the portal, just click on the login now option. Since I already have an account, I'm going to click on the sign in and proceed with it. So once you sign in, you're able to view the various courses and example projects that will be appearing under the Courses tab. So please feel free to browse through the list at your own pace. So since most of the participants here will be interested in precision medicine, just enter the keyword precision medicine, and you'll be able to see the various example projects that will be associated with today's topic. So if the participants are facing any kind of technical issues or having any queries so far on signing up on the portal, please put them in the chat. I'm just proceeding along so that we don't delay the program. So now coming to the part about updating your account. For this, click on the tab that says Welcome Back with your email ID. So how do you update the profile? Like I said, so for that, click on this button that says Profile. So here you will be asked to upload your profile image, your education background, your name with appropriate capitalization. And as you can see here, I've linked my LinkedIn account and my OrcID. So you can also choose to link your Twitter or Instagram or Facebook. And the mentor of the program and the community manager will be tracking your progress under the Activity tab. And if you're completing any courses, for example, right now I'm completing Python course 2, it will appear under the Courses tab. And once you upload any projects, it will appear under the Projects tab. And finally, if you're enrolling for the program, such as the Bioinformatics for Precision Medicine, it will appear under the Programs tab. And finally, if you complete any courses, you will be able to receive the certificates under the Certificates tab. So I hope all of the participants have signed up. And if you have any queries so far, please put them in the chat. Otherwise, we are good to go. We are good to start with the session for today. Do we have any queries? Has everyone signed up? All right, we have it done. All right, so I hope rest of the participants have signed up as well. Otherwise, if you have any queries, please feel free to put them in the chat. For now, I'm passing on the stage to Dr. Raghav to start with today's session. So over to you, Dr. Raghav.
Thank you very much, Sri Gauri. Let me find ways to share my screen until you, all of you, get your registration done. So can you see my screen and hear me?
Yes, sir. Both are working fine.
Okay, perfect. Thank you very much. So welcome to today's webinar on bioinformatics for precision medicine. We know that precision medicine is actually changing the way we understand, diagnose, and treat many diseases, right? That also include cancer. And this transformation of the way that we treat diseases is actually driven by high-throughput sequencing data, that is high-throughput sequencing molecular data that we get from patients for diagnosis and prognosis purposes, and for animal models, to actually understand the disease that we are actually dealing with. And we can also perform large-scale cell line experiments to characterize the responses of different diseases, and specifically tumor also, to several of the treatment options that we have lined up for them. So to compare with precision medicine, traditional medicine uses symptoms to diagnose diseases and drugs to treat these symptoms. But precision medicine aims to turn this approach on its head. Right? Precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability in genes and individual variability in environment and different changes in the lifestyle for each and every person. This approach, or such an approach, will allow doctors and researchers to predict more accurately which treatment and what kind of prevention strategies will work for a particular disease, and in which groups of people it is expected or it is understood to work better. So this is in complete contrast to one-size-fits-all approach, that is in which the disease treatment and prevention strategies are developed for an average person with less and less consideration for the individual variances and individual differences. So after a lot of research, we are poised to enter a new era in the medical practice, where detailed genetic and other molecular information about the patient's disease is routinely used to deploy effective and patient-specific remedies to treat it. We are about to enter the era of precision medicine. So the future of medicine is increasingly driven by the availability of data and the expansion and the standardization of high-throughput sequencing that results in medical practice that is both ultra personalized and ultra precise. Extracting meaningful implications from this data requires deep analytical expertise, and it also requires integrating several of these data, or several of these omics data from different molecules. So let's take a look at what we got here and what drives this change in the modern medicine. The role of bioinformatics in general in the context of diseases is to allow researchers to access, to process, and to analyze and also to visualize several of these kinds of high throughput sequencing data that we will get introduced to over the course of this program. And this program will also introduce you to several examples of how this connection between this omics data and their explanation, or the reason behind the diseases and their effectiveness into several treatment options was done successfully by researchers studying, for example, viral infections like Ebola virus outbreak in West Africa. We will also take examples from COVID-19 pandemic, as well as we can also study microbial pathogens causing, for example, tuberculosis, and development of resistance to several of the treatment options and antibiotic that this tuberculosis encounters it to. We can also consider cancer and their responses to different treatment therapeutic options that we have. And in the end, we will also have options to work with single cell parasite that causes malaria. So we will briefly touch on viral genomics, viral evolution, and gene expression patterns and detection of significant genomic variants, as well as some important aspects of microbiome and virome community in a host that are being studied to find important associations of these several different omics data with disease susceptibility and disease outcomes as we move along. Yeah, my animation is running slow. So cancer, I don't have to introduce all of you to cancer. Cancer is the leading cause of death worldwide, and this accounts for nearly 1 in 6 deaths, right? Some of the most common cancers, as we know, are breast cancer, lung cancer, colon cancer, rectum cancer, and prostate cancer. Cancer-causing infections are prevalent, and they are such as papilloma virus and hepatitis virus are responsible for approximately 30% of the cancer cases in countries that are still underdeveloping or still developing. Right? So many of these cancers can be cured if they are detected early and if they are treated effectively. And this is where precision medicine is applied to enable early detection of cancer by studying various different molecular drivers in these tumor cells and in these tissues that are responsible for the tumorigenesis and for the tumor progression. Is this the case? If somebody else can confirm, can you hear me?
Yes, sir, we can hear you.
Yeah, Ziaul Hassan, could you please check your speaker settings? Thank you for the confirmation. So as we already spoke about cancer, cancer is one area wherein precision medicine seems to be making significant headway, right? And new therapies seek to target the specific cellular pathway that is being exploited by cancer with a view to make something like a short life of tumor, or with a view to make intervention to the tumorigenesis and the progression of this cancer or tumor. So there exists an urgent need to identify new biomarkers or novel signatures that will help us in early detection and prognosis. New biomarkers and signatures can be and are in fact identified by leveraging publicly available omics data resources from the Cancer Genomics Association or TCGA, from NCBI resources, and from various other public resources, which you will get good introduction and good demonstration in this program to find the data and to download the data and to process and analyze the data so that we can find some novel associations with the disease progression. So the field behind this omics data analysis is the big data bioinformatics, and this has developed tremendously in the last several decades, that transformed life sciences and that enabled biotechnology, and also is empowering biomedical research to a completely new facet. So as a result, there came many new discoveries from researchers and from analysis of existing omics data that includes new treatment, that includes diagnostic assays, that design strategies for novel experimentations that will confirm the effects of these identified biomarkers, treatments, and assays. So the main catalyst, as we discussed, for the transformation of the biomedical research is the emergence of sequencing technology. And in this program, we will introduce you to sequencing methods, and particularly next generation sequencing methods or high throughput sequencing technology that will help us study genomic variations, genes and their expression, RNA transcripts and their parts in the disease progression, and how all of these molecular elements are linked to disease biology. So to give you a sense of how this genomics data came into existence, then we already know that it's already been almost 50 years since the time of sequencing of the human genome. And in around 2015, the 1000 Genome Project brought significant number of whole genome sequences to the research community for analysis and for finding implications. That 100,000 whole genomes were sequenced by 2018, and today there exists, or there is an ever increasing number of data sets that are available to help researchers to understand cellular processes, to understand molecular functions, and in the context of disease biology, and in the context of, for example, rare diseases, and also in the context of cancer biology. So this growth of high throughput sequencing data is largely driven by the cheaper, faster, and more efficient sequencing of next generation sequencing, or simply NGS, right? From around $1 billion in 2001 to around approximately $3 million in 2008. And by 2017 alone, the cost of a single sequence genome was close to $1,000. And the aim is to reach around less than $500 or $100 by 2025, I believe, if I'm not wrong. So this has put a tremendous volume of data that is available, genomics data that is available, which is driving the whole big data bioinformatics field forward. So this technology like NGS can be used to explore the roles, the relationships, and the actions of various types of molecules that make up the cells of an organism. And many types of these omics data can be generated using, as we know, right, the next generation sequencing technology. And we will also study, or we will also discuss in this program, how this data can show detailed information about genomic variants and the patterns of gene and isoform expression and the differences in the microbial communities that drive the differences in the disease biology. So the economics of this data are hugely or extremely important for clinical practice and translational approaches of the implications that we can find and associations that we can find. So far, medical data as simply clinical data to introduce you to is a type of information that we have been using for centuries, right? It's like measuring temperature, measuring heart rate, measuring blood pressure, and other symptomatic data that is on the surface. These are typically easily and quickly measured or cheaply measured and are covered appropriately by insurance. But with the advent of molecular biology and cellular biology, and with the next generation sequencing technology, we have learned how health and diseases are driven by what's happening at the subcellular level, or what's happening at the intercellular level. And biomedical data refers to the combination of all of these omics data that characterizes the subcellular level of the disease biology with the clinical and relevant clinical data that is happening at the surface, right? And these different omics data include genomics, proteomics, and even microbiome of a patient that can be related to in the difference in which that these microbial communities help us process the metabolomics that we have in our body. So to give you an example of one of these omics data types and how they are used and how they actually change the approach. So as a method of measuring RNA translation, transcriptomics data or transcriptomics studies can actually help us detect mRNA expression inside a cell as an accurate alternative to measure membrane protein expression, which is actually used, for example, traditionally to, let's say, to characterize tumor histologically. This change in approach to focus on the molecular precision in a molecular level actually changed the precision in, improved precision in diagnostics, and it also enabled development of treatment based on molecular pathways instead of observational chemistry or observational characteristics of diseases, specifically tumor, for example. So the classification of tumor itself has changed, or it actually started to include molecular classification in addition to histological classification or functional classification that one can observe or that one can obtain superficially. And this includes, for example, classification at the level of molecules with complex variation across genomics and transcriptomics data, and also variation that will also include risk profiling of the patients based on the models that can be generated from the data trends that we find from these genomics, transcriptomics, proteomics, and even metabolomics data. And to give you an example, in addition to the existing PAM50 classification and histological classification of breast cancer, we now have a separate set of molecular classification for that characterizes breast cancer in terms of luminal A or luminal B, basal-like or normal-like or Claudin-low, etc. So these are also described, or these are also demonstrated to be extremely important to classify, as several of these different classified subcategories react differently or respond differently to treatment options or therapeutic options. So the significance of the data that we just discussed is huge, as cancer can be diagnosed earlier and the treatment can be selected and adapted as the new data is generated, and the drug safety can be monitored, driven by the complex modeling of the disease progression themselves, not based on the superficial or not based on the what I'm looking for is response related data, but it's based on subcellular and system-wide data. Not based on the symptomatic data, but based on the subcellular, molecular, and system-wide data. That was what I was looking for. So in short, the gene expression pattern, for example, can be associated or can be integrated with the clinical data, and this can help us identify molecular biomarkers for diagnostics and for therapeutic selection. And new discoveries include better understanding of complex diseases at the subcellular level, as well as it can help us in the target identification for novel drug design. It can also help us repurpose existing drugs for different purposes. One of the very famous example is the usage of metformin drug, which was traditionally, or which is still used to treat diabetes, is actually getting repurposed to treat clinical cancer data, or repurposed to be anti-cancer drug in certain conditions based on the omics data analysis. So what this actually creates is, this creates a disconnect between, for example, the data that is obtained from the lab technicians and clinicians, and the data that can be processed and understood from the biomedical research ecosystem based on the data analysis method and data analytics that we can perform using bioinformatics. This disconnect actually breaks up the cycle of innovation due to lack of data-driven research skills among the otherwise very effective biomedical personnel that we have in dealing with increasing demands and increasing changing in the unpredictable landscape of the omics data that is currently present. So to fill this gap, what we need to do is we need to develop a more for example, ecosystems that are prone to be decentralized and that are prone to be data-driven approach, right? And we have been given a taste for this current scenario from thanks to the pandemic, or not thanks to the pandemic. This has already started to affect healthcare, and more subtle trends of disease management and drug development are also being observed, right? And these industries are most affected as going decentralized for laboratory has been found very difficult, and several harsh COVID protocols has to be taken to enable working inside this laboratory environment. But on the contrary, data-driven approaches in large-scale projects can be performed in a decentralized manner, and this is key to the future of clinical and biotechnological innovation, where we have plenty of omics data, we have plenty of clinical data, and they are waiting to be approached in a data science oriented or a data science related approaches. And in this program, we will ensure that you get a taste of how this is done by taking data from different sources And then we will perform analysis on the server associated high performance computing server, the server powered Bioinfo server. And then we will show you how to make the associations from the omics data to the disease biology based on our analysis on this data. So we will also learn about basic and translational research in, for example, oncology, where the patient-derived cell lines or xenograft models and clinical trials offer a rich collection of data to for example, help us treat cancer in patients and help us give informative diagnosis at the patient level. So this, as I mentioned, has completely changed, and it has given us a complementary information approach towards disease biology, where we have traditional biological hypothesis-based approach can help us validate once we have identified specific markers and specific therapeutic responsive agents, elements that we can use in the traditional approach to verify and traditional approach to build extremely targeted assay-based methods to understand different, for example, understand personalized and precision medicines reach to cancer patients who are responding differently to different treatments. So how do we go about this? For example, as I already introduced you, so we have to analyze, and we have to, for example, consider data from different sources, like from different molecular sources, from different even different sample sources like microbial composition of our bodies and the environment that it is actually thriving in, and how they are responsible for destabilization of the immune system, and that leads to clinical manifestation of disease specific to the microbial abundance in specific parts of our body. And we can also relate or integrate cellular omics data with clinical phenotypical data, which is done at several different integrative levels that can be associated with common diseases, and that can also be associated with specific and rare genetic diseases, and that can be associated with cancer also in many different situations. Right? So, do we have promised to develop new methodologies and new informative approaches to data that is already been present, or do we have to resort on generating new data? But we can see from this graph that there is an information explosion, and there is an increased reliance on the data due to the lowering of costs that generate these detailed and large data. So, however, there is an ever increasing or growing information lag that complicates and the translational approaches of this data into practical and impactful insights at the point of care. So this is where, or this is what we call the analysis gap, and this is proportional, or this depends on our ability to generate new data and our ability to process, digest, and analyze the existing data to interpret it for the benefit of the individual patients. So existing data, such as data repositories from the Cancer Genome Atlas or data from NCBI, can be used to find novel approaches and can be used to find novel biomarkers, or can be used to find biomarkers specific to specific ethnicity and race and demographic classifications of data. So what we actually address in this program is to, wait a second, So what we want to address in this program is to close the gap by focusing on the central issues that define the precision medicine, right? We will offer you training, support, and effective tools to manage, process, and analyze, and also to integrate the data so that we can make a lasting impact, and we can find novel associations, and we can at some day change the future of medicine. So our first headache or our first challenge is to what do we do to analyze this data? In order to analyze this huge volume of data, one has to resort to the methods, or one has to address the computational complexity of analyzing this humongous volume of data. For example, a genomic data of a single cell can reach the sizes of tens and tens of GB, if not hundreds of GB, right? So what do we use to do, or what do we use to perform analysis on these data? Right? And this is just one of the major challenges that is related to, for example, omics data analysis, to accurately and to effectively process large-scale data sets that represents gigabytes, sometimes even terabytes of data. So such large quantities of data can actually prevent many of us to gain practical and hands-on understanding and hands-on feel for what this data represents and what are the patterns of this data, right? So let us quickly have a demonstration, or quickly have a hands-on overview of various methods of analysis for example, to help you overcome these major limitations. So this is how we will do this also. Let's go back. Yeah, this is one of the addition, or this is one of the pipelines that you will set up in order to understand, or in order to associate transcriptomics data with different tumor subtypes, specifically considering breast cancer in this case, right? You will get access to hands-on exercises. You will get access to the data sets that we used for practice, and these data sets are curated so that the analysis will run quickly. And you will also get access to where we got this data, to the publication that from where we got this data. And these data and several steps that involves in processing this data. For example, we have to pre-process this data. We have to perform the analysis by mapping this genomics data or transcriptomics data to the reference genome, human reference genome. And in the end, we have to identify variants in our data, or we have to identify, or we have to quantify the expression levels of different genes by including appropriate analysis methods for setting up the pipeline, let's say for transcriptomics data analysis or for genomics data analysis. So one of the results that you will get is such as this, where we can identify genes that are differentially expressed in the diseased state compared to the normal state or normal tissue. This can also help us identify genes and pathways that are enriched, for example, in a tumor tissue compared to the normal tissue that is obtained from the same patient, or in a tumor tissue and data from normal tissue that is obtained from healthy individuals. So we will learn about some of the important projects and some of the data that it collects, and there the Cancer Genome Atlas is one of the very important repository that has a collection of large quantities of several different omics data for over 30 tumor types. We will get a detailed introduction to navigating this repository, finding important analysis, finding important data from this repository. We will download relevant data from this repository and perform analysis pertaining to precision medicine to understand how this repository can be leveraged to perform a project in precision medicine, let's say. On the other example, or on the other hand, we will also deal with data from patient-derived cell lines. So these are data that are generated from under one roof or from a laboratory, where they consider several numbers of patient-derived cell lines and characterize these cell lines for multiple omics data types. So the advantage of using a model system like a cell lines data over patients' data that is observed, or that can be obtained from Cancer Genomic Atlas, is that these patient-derived cell lines can be subjected to several analysis, several experimentation methods. For example, in this study, these cell lines that are derived from the breast cancer patients are actually first characterized at different molecular level and also studied for their responses to 90 different drugs, right? So now we have data that characterizes these cell lines or tumor at multiple molecular omics level, and also we have data of their response to several different treatment options. So by integrating these two data, we will get how we can effectively identify specific molecular characteristics that can be associated with a positive or a sensitive response to a therapeutic option, or negative or resistive response to several therapeutic options. Then we can use these biomarkers and apply that to patient data from TCGA, for example, to identify or to stratify patients who are most likely to respond to the treatment options that we have identified from this study. So this example project will also be dealt with in tremendous detail in the upcoming program. So this is, for example, if we have to identify and associate the omics data to the clinical data, this can be done using data that we download from the Cancer Genome Atlas, as we have been going through. In this case, we have an example project that deals with liver hepatocellular carcinoma and how various clinical data available on those patients, for example, their risk factor and their demographic information and their, for example, disease history in terms of hepatitis virus and their complications, can be associated with the molecular characteristics or molecular omics data like mRNA expression or gene expression data and microRNA expression data. So these association and integration can be made in this project, and this will also be discussed in detail in this program. So we will, in addition to this, look at, for example, phylogenetic tree reconstruction to understand the spread of Ebola virus outbreak in, for example, around 2016. And because we don't want to focus only on the cancer, we want to explore the precision medicine approach to infectious disease research, where this specific branch of precision medicine is fastly improving and are being applied to several different disease states. So another example that we will consider is to study SARS-CoV-2 genome and the cell's responses, as well as we will see how metagenomics or microbiome data is associated with the disease and drug response. So these are also we will consider in this program. So for different parts, we will this been already covered by Sri Gauri, but we will leverage the omics logic resources. Go back again. It's running or it's not running. Yeah. So we will leverage these resources. For example, we will use asynchronous coursework. We will use the cloud-based analytical server where you can analyze large genomics data. And we will use, for example, code-based lessons where you can also practice coding in R and Python. And we will also use several of the project examples that uses published data or that uses data from publicly available repositories. So to proceed further, these methods, let me actually give you an introduction to some of the pipelines or some of the outputs that you can take from the server to successfully analyze omics data into multiple different analysis pipelines that is going to give you information on the phylogeny, that is going to give you information on the gene expression pattern. It will also give you information on the structural and functional aspects of the variants that you identify from variant calling methods. It will also give you, for example, help you perform surveillance-based approaches and surveillance-based analysis on publicly available resources for for example, for SARS genome, for a SARS-CoV-2 study. So let's, without further ado, let's go to server and let me show you how you can perform this analysis by setting up a very simple demo pipeline for transcriptomics data. So let me reshare my sharing. Okay, just let me know if you can see my screen once I set up the sharing. It's still working. Yes, can you see my screen? Can you see the server page?
Yes, sir, we are able to see.
Okay, so if you go to the server Bioinfo, this is a page that you will take a look at. So once you have some credentials set up, so this demo pipeline can be accessed by everybody who has a credential in server Bioinfo, server in the big data server. So you don't have to be a paid user to analyze, to perform this demo pipeline, which will give you an example or experience of setting up this pipeline. The data that we are going to consider is a cell line data that I have been talking to you about. So that analysis will come under the transcriptomics data analysis. And you can, for example, to set up the pipeline, you can drop down this box and then choose the option demo PDX project, or sorry, let's go here, so not the PDX project, demo cell line project. So if you click this, this is going to open up into the demo pipeline. And yeah, so we will start by starting. So this has the input files have already been uploaded. So you just have to follow the lead of the highlighted sections that this demo pipeline is offering you. And the choice of this analysis method, you will get to know as you go through the program. So first you have to do the pre-processing. Once you select these analysis options, you will get suggestions, right? I'm getting now suggestions based on what could be my future step. So I want to perform mapping onto the genome using HiSat2, and then identify some novel isoforms based on my sample, and their expression should be quantified. And when we do this, I will want to quantify the expression levels of the isoforms and genes I have mapped onto the genome. And then I want to perform a downstream analysis or differential gene expression analysis, which will help me identify, or which will help me shortlist genes that are differentially expressed in one type of cellular or molecular data, molecular tissue type compared to the other. So I will end the pipeline. And then by ending the pipeline, you will get hands-on approach, or hands-on to several of the output files that you can download. For example, we will have expression levels of isoforms and expression levels of genes that are at different normalization, that are performed for different normalization. And as I also mentioned, you will get statistics of how well your FASTQ files or the reads mapped onto the human reference genome. So this will become the quality control of your mapping. And we will also get results of our differential gene expression analysis that we have performed by comparing cell lines or by comparing tissue types that are normal-like compared to the clotting load. To perform this analysis, you need to have prior biological information about the sample. So this is how you can set up this analysis, and you can access these output files. And if you download this zip file, you can have access to all the plots that differential gene expression analysis DE-Sec2 produces, so that you can make sense of all the data and make sense of all the outputs that you have obtained by performing these omics data analysis and downstream analysis in the server. So I urge all of you to explore this part and explore the demo pipeline of the server. And with that, I will stop my presentation and open the podium for some discussion. And if there are none, I will be happy to hand it over to Sparsh. And if you have some questions, please let me know in the chat.
Thank you, Dr. Raghavendran. Sonalika, can you please make me co-host? Hello, Sonalika, am I audible?
You are audible, Sparsh.
Let me try to share my screen if I can. Is my screen visible?
Yes, Parsh, it's visible.
Thank you, Ayushi. So as I'm not the co-host, it would be great if Ayushi or Shubhar, you can share the appropriate link in the chat box while I'm discussing or telling about the registration part. Thank you. So, hello everyone. I'm Sparsh and I'm the marketing manager here at Biomedical. So I'll be just quickly explaining you about the registration part of this particular program, that is Bioinformatics for Precision Medicine. So as you can see that the program has been commenced from today onwards. So today was the first session of the program, and this is the program page. When you scroll down, you can see the program overview. In this, you will get to know why this particular training program, mentor guided training program, has been designed, for whom it is designed, what are the sessions or the topics we are covering, what are the practical aspects which we are covering in this particular program. If you scroll down further, you can see and elaborate a comprehensive, detailed curriculum of the program. And as today was the first session, the session was on Introduction to Bioinformatics for Precision Medicine, where Dr. Raghavendran has covered all these topics, that is data analysis, combining clinical and molecular data, from the lab to the clinic, research, discovery, and clinical uses cases, case studies in precision medicine, etc. So the next session is on October 10, and it will be on genomic data analysis, where we'll be covering about the NGS, genomic data, NGS data, analysis of NGS data and variant calling, annotations of variants, identification of clinically relevant mutations, etc. So as you can see on the right hand side, or that there is an interactive session, a calendar, and that particular calendar, you can see some of the dates are highlighted. So these are the dates when we'll be having our next few sessions. So the structure of the program is quite a hybrid kind of, that either some of the session would be asynchronous session, some would be a kind of a live Q&A or live session. So we will tell you and we'll mail you regarding the same accordingly. And whosoever will be like enrolling for this program and will be the participant of the program. And now, apart from genomic data analysis, we'll be having sessions on transcriptomic data analysis, then phylogenetic analysis and MSA, our response to infections, precision medicine for neurosciences, metagenomic data analysis, advancement in precision medicine, and oncology data sources, raw data and public databases, and so on. So these are the topics which we'll be covering. So you can go through with the these are the sessions which we'll be covering, sorry, and you can go through with the topics like in those per session, which we'll be covering further. Now, when you scroll down, you will see 3 different tenures. One is of 45 days, another is of 60 days, and then is of 90 days. So the common resources one would get if they enroll for this particular program is online tutorials, project examples, coding sessions, session recordings, online mentor support, and online technical support via our Skype community. Then certificate of completion and example cloud pipeline on T-Bioinfo server. Now, what's the difference between the three is in 45 days tenure, you will have the access to only the online mentor support and online technical support. And in 60 days and 90 days, you'll be having few more opportunities. In 60 days, you will be having access to the educational cloud pipeline, the educational license where you can upload your data set, which is being present in the public repository. Secondly, you can have access to the one-on-one mentor sessions. So whenever you get stuck somewhere while going through your training part, you can fix a kind of exploratory call with the mentor of like 15 to 20 minutes and discuss and clarify your doubts there. Now, second thing is you have an opportunity to draft a research proposal, and that particular proposal you can send it to the mentor for evaluation purpose. So mentor will evaluate your research proposal and guide you accordingly. So whatever amendments or changes that needs to be done so as to make the research proposal more validated, he will guide you accordingly. The outcome for 60 days is a poster presentation in a research symposium or any other international or national conferences. Now for 90 days, with one-on-one mentor sessions, online mentor support, online technical support, you will have an access that you can prepare and or you can work on your own independent research project. And the outcome of 90 days tenure is that you can have a research publication in a very good peer-reviewed journal. So this was about 45 days, 60 days, and 90 days resources. Now talking about the pricing part, the fees part. So here you can see that there is a drop-down kind of menu, and you can change according to your location the currency. For now, I am using USD dollars. So the pricing for 45 days is $150. For 60 days, it's $300, and for 90 days, it's $450. And we do have scholarships. So I guess Shri Gauri can share the details of mine, like the email address and the official contact number, which is active on WhatsApp. And you can further contact me. You can connect me via WhatsApp, and we can discuss further about the scholarship, how much we can avail. So this was a brief about the tenure, about the registration and the pricing part. So if anyone is having any doubt related to the registration part or the scholarship, please feel free to put their question in the chat box, and I'll be there to answer it. Thank you. Over to you, Shri Gauri.
All right, thank you so much, Sparsh. So does the participants have any further queries? I've shared Sparsh contact details in the chat. So if you have any queries, please feel free to get in touch with her via email or her WhatsApp number. All right, so I hope all of you were able to learn something from today's session, and I hope to see you all for the second session of today's program. With that, thank you so much for your patience and have a great day. Thank you.
Thank you, Shri Gauri. Thank you, everyone. Thank you, Dr. Rao.